In this video, we're going to talk about how the Hardy-Weinberg theory was derived and how to solve Hardy-Weinberg problems. So before we start going into how to solve a Hardy-Weinberg problem, we're going to talk about how the principle was derived. When there are two alleles of a gene, there are three possible genotypes. Homozygous of each of the two alleles and heterozygous for the other two. The frequency of two alleles in a population are represented by letters P and Q. If random mating occurs in the population, then the chance of inheriting two copies of the second allele is Q squared. Therefore, the expected frequency of the heterozygous genotype will be 2 times P times Q. Then the sum of all the frequencies are going to be one new species or 100%, depending on if it's a genotype or a phenotype. The genotype are the set of all genes, and the phenotype is the physical expression of the trait that we are able to see. There are also 5 assumptions made when the Hardy-Weinberg equation is used. The 5 assumptions are no mutations, random mating, no natural selection, an extremely large population size, and no gene flow. Here at Punnett Square, I am going to show you how the equation was created. So say you have a male and female. They both have heterozygous traits. So capital A and lowercase a, and the female also has capital A and lowercase a. Before we start the Punnett square, we gotta remember that capital A equals P and lowercase a equals Q. So we're gonna start off here, and we're gonna multiply capital A and capital A, which is gonna give us two capital A's, which will be P squared, because A equals P. Capital A equals P. And then we're going to go over here and multiply capital A and lowercase a, which is going to give us the heterozygous trait, and that's going to be P times Q. This is going to be the same thing, capital A times lowercase a, P times Q. And then we're going to multiply the two lowercase letters, which are going to be lowercase a and lowercase a, Q squared, because if we look back up here again, lowercase a equals Q. When we add these all up, these are going to be 2 times P times Q, because there are two heterozygous traits. And therefore, that's going to give us P squared plus 2PQ plus Q squared equals 1. And that's how we get our equation. So here's an example problem. There are two alleles, capital A and lowercase a. A is dominant and lowercase a is recessive, which represents a dominant and recessive trait. Calculate the genotypes and phenotypes when capital A is 0.35. So in order to solve the example problem that was given to us, we have to calculate the genotypes first in order to get our phenotypes. So as mentioned before, A equals P and lowercase a equals q. And they give us capital A equals 0.35. Because we only have one capital A, we have to square it in order to get both capital A and capital A. So it's going to be capital A equals 0.35 squared, which gives us 0.1225. Now we need to be able to find our Q value. So Q is going to be 1 minus 0.1225, 35, which equals 0.65. Now we have to find our recessive. So little a is going to be squared as well. 0.65 squared, which is going to give us 0.4225. Now that we have both homozygous genotypes, we need to find our heterozygous trait. So we're going to multiply 2 times P times Q, and that's going to give us 2 times 0.65 times 0.35, which equals 0.455. And these are all your genotype calculations. So we want to be able to check our genotypes to make sure they all add up to 1. So remembering the equation, all of the values need to add up to 1 by adding p squared to pq and q squared. So we are going to substitute all those values with the values that we figured out from before. So we're going to put in 0.35 squared plus 2 times 0.35 times 0.65 plus 0.65 squared, and we're going to make sure that equals 1. When you check in your calculator, this all should equal 1, or close to it. Because the phenotypes are something individuals are able to see, in order to calculate these phenotypes, we need to add capital A lowercase and capital A together in order to get our dominant phenotype. So we're going to get that from the calculated genotypes that we've already calculated from before. So as remembered, capital A lowercase a equals 0.455, and capital A equals 0.1225. As I just said, in order to get the dominant phenotype, we need to add these up. So it's going to be 0.455 plus 0.1225, and that's going to give us 0.5775. And you multiply that by 100, and that's going to give us 57.7%. And that is your dominant phenotype. The reason why we use both of those is because the capital A is going to always dominate lowercase a, and so that is why our phenotype becomes dominant. Now we are going to find out the other part, which is just a recessive phenotype. which is just going to be 0.4225 times 100, and that's going to give us 42.25%. To make sure that these add up to 100%, we are going to add them up. So it's going to be 4.225 plus 57.75. And when you add this in a calculator, it should equal 100%. And that's how you know that's right. So that is how you solve a Hardy-Weinberg equation. Thank you for watching.